Okay, so I'm now going to take you back to medical science or somewhere close to the theme that we started on this evening. But we operate, I guess, one would say, at the right at the cutting edge of medical science in terms of developing advanced therapeutics and doing that as a research exercise from which ultimately we hope and expect to develop therapeutic products. There you go, there's our disclaimer. I'll move swiftly on beyond it. Just a quick sort of bit of background to the company for those of you that don't know anything about us. We're an AIM-listed biotech company. We are involved in developing cell-based therapeutics, pure and simple. So we operate a classic pure-play biotech model. So we raise money from shareholders, deploy those funds into research and development, and ultimately, we hope, generate positive clinical data. And the better those data are, the higher value those programs become. And ultimately, our ambition, in line with any other biotech company, is to license or have those programs acquired by pharma companies in due course. But for us, because we're involved in advanced therapeutics, essentially we will need to take those products further than perhaps would be the case with more conventional small molecule or biologics. Because we are, in many cases here, treading paths that haven't been trodden before. So that raises risk. It also raises potential returns at the other end because we're focused on unmet medical needs primarily. But we need clinical data, and clinical data at quite an advanced level in order to make those programs unequivocally attractive to a potential partner or acquirer. So it's always been important for us to really give ourselves the financial capability to take these programs to the point at which they will be valuable, for better or worse. Now, there are some things we can't predict in terms of the clinical data, and that's the nature of the business that we're in. But what we try and do with these programs is stack the odds as far as we can in our favor, and I'll try and explain how we do that with the programs that we're currently working on. We're targeting primarily major unmet medical needs. We have programs, all of which are now in their clinical phase, addressing stroke disability, the consequences of stroke, critical limb ischemia, which is a common side effect of type 1 diabetes. We also have a program looking at blindness due to degenerating retina. That's actually an orphan status disease, so it's a smaller market, but that's also highly attractive in terms of speed to market because it's a smaller population and inherently orphan drugs offer a faster route through the regulatory system and typically focus on smaller populations, and that also means the prices at the other end can be that much higher. But I'll come back to all of these programs individually as we go through them. If there's time at the end, I'll also try and explain a little bit about a new platform we're developing, harnessing a byproduct of our main platform stem cell manufacturing process, and that is the exosomes we can harvest from those stem cells, which is taking us into a new area for us of treatments for cancer. So before we get into the programs, again, just a quick word on shareholders. So we're fortunate to be backed by, I suppose the best way of describing these investors is patient capital. And indeed, Neil Woodford's funds includes both the Patient Capital Trust, actually also the original income fund that he originally set up. And obviously, I guess Neil's known for being one of those patient investors who is willing to take bets on biotech companies which meet the criteria that he and his team look for. We also have a number of other key institutions, and quite a number actually beneath the notifiable threshold as well. And that's important for us because we need these investors, A, to understand what we're doing, and to have the patience to allow the story to play out over time. What that's given us is a good deal of money on the balance sheet. Our market cap is a good deal less than 100 million at the moment, and as you can see, a large proportion of that is actually accounted for by cash on the balance sheet. And that is important because we want to be in a position to be able to pursue these programs at clinical level, undertaking clinical trials which are expensive once you get to phase 2 and beyond, undistracted, without the need to have to go back to the market with a looming milestone that's forcing you into a corner. So now we have cash runway right out actually to the end of calendar 18 and possibly a bit beyond actually, depending on how one measures it, to be able to get the job done. And I'll come back to the milestones towards the end that we intend to achieve, primarily clinical data milestones using the cash that we have on the balance sheet there. So why are we engaged in stem cell research and development? Well, what we're trying to do as a business and what we've been working on for the best part of 2 decades now, which gives you some indication of how many boxes one has to tick, both scientifically and then clinically, to convert stem cell science into therapies. What we're trying to do is harness the power of stem cells per se, the power of stem cell biology. So we all have stem cells, they are nature's repairing cell, and the great potential of stem cell science, in theory at least, is to offer a new paradigm for treatment of hitherto intractable diseases, to offer even some curative potential for these conditions. So there's a great prize to be had here, but the big problem is how do you convert stem cell science into therapeutic products, something that can be made, scaled, and sold at a price that healthcare systems are going to be able to afford. And it's those issues that are coming to the fore now in the broader field of stem cell development. The science is all very well and all very exciting, but for us, what's equally important to us, and certainly more so actually these days, is how one actually is able to do the boring stuff, to be able to make these stem cells in a GMP facility at scale in a consistent way with release criteria that a regulator will understand, and to be able to do that time and time again at a reasonable cost of goods that a licensor is going to, or the licensee rather, is going to be able to make a decent margin on, and which healthcare systems can reimburse ultimately when they get to market. So, so much for stem cell science. What are we actually working with? Well, we've developed 2 platforms over the years. Our platform called CTX was the one we've been working with for over a decade. It's a neural stem cell line, so it was originally derived from brain tissue, and what we've done is genetically modify an original founding cell to immortalize that cell into a cell line. And every cell within a cell line is precisely the same. So it gives us a very well characterized product, if you will, or therapeutic candidate from which to develop therapeutic treatments. So we're using CTX, this neural cell line, as the basis for our treatments for stroke and limb ischemia. And over the last few years, we've also developed a second cell line called HRPC. That stands for human retinal progenitor cells. And this is a population of cells in this case, non-modified to expand it. We actually expand it in low oxygen culture. Why do we do that? Because that's actually the environment you get at the back of the eye. So we thought that might be a good idea, and it actually did work for us. And those particular cells were derived again from tissue from the back of the eye originally. So we're trying to use tissue that is originally derived from the organ of origin that we're actually interested in treating diseases that are associated with that organ. So just going back to the notion of how do you convert stem cell science into a product, over many years what we've been able to do with CTX is to kind of tick the boring boxes. We've been able to manufacture it at scale in a GMP facility, which we're now building out actually in South Wales, having relocated the business down there. The Welsh Government's kindly deploying European funds to build that out at their expense, and we'll become a tenant, well, we are actually a tenant now of that facility, and the manufacturing part of it is being completed. The research and office part of that facility is already operational. We're all down there. So we have the means to be able to manufacture this product. We can also deploy it with a long shelf life. So we can cryopreserve this cell line, put it in a cryo shipper, that little bottle thing you see on the bottom left there, and that means we can store it, ship it, and it can be deployed and taken out of pharmacy in the vast majority of hospitals or hospitals that are going to be treating patients that we're targeting will have the ability in pharmacy to store cryopreserved product, to thaw it, dilute it if necessary, and then administer to the patient. So what we're trying to do here is to get our stem cell therapeutic candidate as close as possible to a more usual biologic paradigm, drug paradigm, that hospitals have been used to using for many, many years. And with CTX, uniquely, we believe we've ticked the boxes that are necessary. What we now need to do is generate the clinical data with this product to show that it is effective or effective enough to become a product in market. Our pipeline is listed here. Every good biotech must have a pipeline, and here's ours. What you see in blue is where we've got to currently with these programs. So CTX for stroke, for limb ischemia, the HRPCs for retinitis pigmentosa, which is our first retinal disease target, and more speculatively, our first exosome-derived candidate for glioblastoma, brain cancer. The next phase is where we're looking to take these programs with the money that we have in the bank, as previously mentioned. Before I just briefly get into the programs in turn, a quick nod to the management team and our non-executive board. And we've built this out over the last 18 months to 2 years to really give ourselves the bandwidth we need in all the key operational areas that we're going to need to address in terms of medical, clinical, regulatory, and so on to get the job done. So there's a good team of people. We're about 50 people in total, and with our manufacturing facility in South Wales, that will probably rise to no more than 70 to 75. So we have no ambition to become a 500, 1,000 person company. We can get the job done with those programs with less than 100 people. And clearly, although we have a fair bit of money in the bank, keeping an eye on the cost base is clearly very important, especially as we are essentially a cash burn company, not currently making revenues. So let me just go into the programs briefly in turn. I'll start with the retinal one. And the reason I'll start with that is it's actually our first program that we got into a US clinical trial. So this one went straight to a phase 1/2 study in the US. So we're delighted finally to have got our reach, if you like, into what is obviously going to be a very key market to us, and through obviously a very, very rigorous regulator, the FDA. RP, retinitis pigmentosa, is a degenerative disease of the retina. What you get is what you see in the image there, this gradual loss of peripheral vision, which over many years, in some cases, will ultimately lead to blindness, so a progressive loss of sight. It's an orphan status disease, as I mentioned before, so the overall population there is about 250 to 275,000 patients in the US and EU combined. So probably on the orphan side of things, quite a large orphan indication. But nonetheless, it meets the criteria, and that gives us the ability to get orphan drug designation, which helps the regulatory pathway. We have that both here in Europe and in the US. And indeed, we have fast track designation for this program from the FDA as well in the US. And again, all of these things will help swiftly move this program through its clinical development pathway, obviously assuming that the data is good enough. So we have a phase 1/2 study running in the US at the moment. It's a relatively small study, as most stem cell-based or advanced therapy trials are. They don't need to be very large studies because the needs are unmet, there are no comparable treatments, et cetera, et cetera. So here we're running a brief safety part to this study, 3 dose cohorts of 3 patients each. And then we'll go on and treat 6 further patients at the highest safe dose from the phase 1 element. Primary endpoint, obviously safety, but we'll also be monitoring efficacy measures, visual acuity, and so on, as those patients follow up over time. Obviously, you've got 2 eyes, so we have a natural control there, which is great. The eye is a very nice organ to work in if you're a surgeon, or especially a retinal surgeon. Because it's a closed system, it's a really nice, straightforward organ to be able to work with. And the image there looks rather gruesome. But if you're a retinal surgeon, this is relatively standard stuff. The cells are injected to the back of the eye in the subretinal space by way of subretinal injection. So something that's fairly routinely done for other types of retinal-based treatments. We're working with the Mass Eye and Ear Infirmary in Boston, one of the world's leading centers for this type of work, under Eric Pierce, who's one of the US doyens in this area. So we're delighted to be being able to run this study at a center of that, with that, or as prestigious as that. I'll come back to the milestones and the timings associated with these studies in a few moments. Just moving quickly on to stroke, or the consequences of stroke, as I mentioned. Stroke is the single largest cause of adult disability in the world. It's a very, very significant unmet medical need, and the social and healthcare costs associated with treating disabled stroke patients are very, very significant. And of course, this one is also playing into an aging demographic. So these numbers are only going to get larger, unfortunately. even though in certain territories or some of the underlying causes of stroke are being addressed, i.e. reduction in smoking in the developed world, there are other factors that are taking it the other way, most notably obesity and diabetes and the like. So we have spent many, many years developing a treatment to address the consequences of stroke. So we're not looking to try and save patients in the acute phase, which is where virtually all of the conventional drug treatments have been tried, most of which have actually failed. What we're looking to do here is to take patients that have stabilized from their stroke but have been left with a residual disability. And generally, if you're going to survive a stroke, and a third of patients unfortunately won't survive, and half of the survivors will have a residual disability, that's going to be manifest in around 30 to 60 days, and in the vast majority of patients it won't improve thereafter. And by disability I'm talking about paresis, so basically an inability to move on one side of the body due to an insult in the brain, an ischemic stroke on the other side. So we're looking to really ultimately make these patients be able to move around a bit more easily, thereby lead a more independent quality of life and be less reliant on health and social care. And that's where the economic argument is for this intervention. We've already run a phase 1 study in the UK out to 24 months follow-up in 11 patients. This was published actually in The Lancet earlier on this year in the summer. We saw no cell-related or immunological adverse events, so the treatment has a very good safety profile, it seems. And we did see encouraging efficacy measures against these patients' baseline level of disability. And those patients were tracked for 10 weeks prior to getting the treatment to ensure they did have a stable level of motor deficits, which were then measured over time out to the 24 months. Where we saw improvements, they tended to be manifested around 3 months and were then sustained right the way out to that 2-year follow-up point. So encouraging, and has led us into a phase 2 study, which is currently running here in the UK across 9 or 10 hospitals up and down the country. We're using the highest dose that we used in phase 1, 20 million cells, injected directly into the brain, which is an overnight procedure for the patient, so they'll typically go home the following day or the day after. And the primary endpoint in this study speaks to what I've just described, which is this ability to move the body post-treatment, especially the upper limb, because one can compensate for lower limb disability by limping or swiveling the hip. It's very difficult, though, to compensate for upper arm movement. And if you watch the Andrew Marr program on a Sunday morning, you'll see that his left or his right arm just sits there and doesn't move. And that's a classic post-stroke motor deficit that he's suffering from. So what we're looking to do here in this phase 2 study is measure a treatment's ability to perform a useful task, in this case to lift an object from desk height to a platform, pre and post-treatment. It's a very, very clear readout of whether or not this treatment is effective in addressing motor deficits in these patients. All being well, and those data, by the way, will be available this December, so we've treated the patients, they're now in their 3-month follow-up period, and we'll be reporting initial data from this study in December. And all being well, that will lead us into a controlled phase 2/3 study next year using the totality of the data we've already garnered from phase 1 and phase 2. In the controlled phase 3 study, we'll be using placebo surgery. So those patients will actually be monitored against a true placebo group that will get the surgery but not the cells. Clearly, when one's doing placebo surgery in the brain, it raises all kinds of regulatory and ethical issues. So for us, it was very important for us to be able to show a clear efficacy endpoint in phase 2 in a non-controlled setting before we wanted to take this treatment to regulators for a placebo-controlled study. And that's pretty much where we've got to. So just a quick word on CLI, because I'm running out of time. Critical limb ischemia is a loss of blood flow to the lower limbs. It's very common in diabetics. Ultimately, this will lead to amputation, initially dry gangrene, which is what you see in the rather horrible image there. And what we're looking to do here is deploy CTX and its very, very potent ability to regenerate blood vessels, especially in the microvasculature, as a potential treatment to avoid amputation in these patients. We're currently in a phase 1 study in the UK that will read out again by the end of this year, and all being well, that will lead us into a phase 2 efficacy study next year in this indication. And because I've only got a minute left, it seems, I I'll gloss over the exosome platform. Suffice to say that it's a grant-funded, in this case, early-stage program that we'd hope to get into the clinic maybe next year, back end of next year in brain cancer, based on encouraging early preclinical data. And as I mentioned, we can harvest these exosomes as a pure byproduct from our CTX cell expansion process, and then purify those exosomes as a therapeutic agent in their own right. In terms of the milestones, these are all clinical data driven. So what you see here are the clinical starts and data readout points over the next couple of years or so across the 4 programs I've mentioned, starting with CTX in both stroke and CLI, then the retinal cell line in RP, going from phase 1/2 into phase 2/3, hopefully over the course of next year and into 2018. And more speculatively, the exosome product at the end there, if we can get that into the clinic next year. So these are milestones that, if those data are positive, will generate value in this company and ultimately make these programs licensable. So just to sum up, we have differentiated stem cell technologies. These are scalable and cost-effective. There are clearly very high barriers to entry with this type of endeavor. We're targeting very significant diseases. There are unmet medical needs. We can offer a clinical portfolio with multiple clinical readouts over the next 2 to 3 years, and we're funded to achieve those with a strong institutional shareholder base, a good leadership team, and very significant opportunity for value build, especially based on the current market cap, which frankly isn't that much higher than our cash balances. So if we can get even just one success with one of those clinical programs, our market cap will reflect that, and then some. That is the nature of the biotech model that we're running. And with that, I'll leave it. Thank you.
Questions, anybody? I knew you would.
Michael, you've got quite a short shelf life of 6 months. How do you assign that? Is there an assay which you do to test the cell viability?
Yeah, and I mean, actually, when we did the phase 1 study, it had a shelf life of 8 hours. So we were manufacturing in Edinburgh, shipping to Glasgow. It was a single-site study within that shelf life. So actually, 6 months in the scheme of things is quite a luxury for us. We can extend it far beyond that, as you might imagine, with a cryopreserved product. In theory, there's no reason why it shouldn't be much longer than that. But all that will come with data, basically stability data, that we garner the longer and more, the more we use the product and the more we manufacture. Certain amounts are set aside and we monitor for stability over increasingly long periods of time. So 6 months is really where we've got to thus far, but only because that's the only stability data we have so far. It's
What sort of assay test is it then? Is it just an examination, or are you actually trying to multiply the cells?
I mean, we have pretty extensive release criteria for the cells. So you're monitoring for viability, or you're monitoring for potency against cell surface markers and the like. So none of these are particularly esoteric within the scheme of stem cell science generally, but they are fairly conventional assays to deploy. And frankly, the regulator will want to see something that demonstrates that you can identify the cell as being whatever it is, a neural stem cell, and then something that demonstrates that it has potency. So you have to have assays that measure that. And all we'll do is monitor the cells to ensure that over time, those cells meet those assays or those release criteria, at the point at which they are measured.
At some stage in the future, when you have regulatory approval, do you envisage that hospitals will be ordering a batch of these cells for each individual patient, or will the hospital pharmacy freezer be full of all your product just waiting to go?
It depends a bit. I mean, I think in the case of stroke, it depends on the centre. There are stroke centres that will have a steady stream of patients, many of whom won't be eligible for this treatment because they either won't survive or they will have the nature of their stroke, because every stroke is different, it's a heterogeneous condition, will mean that they will have a disability that is either more or less amenable to the treatment. So we estimate that probably maybe around 10 to 20% of stroke survivors will be eligible for this. So there'll be a certain amount that won't. And actually age will also be a factor as well. So from a pharmacoeconomic standpoint, and it sounds fairly gruesome to say it, but the older you are, the less you have to lose from a pharmacoeconomic standpoint. So if you can get a fairly early, perhaps first stroke survivor back into economic activity, that's really where the numbers start to become dominant in the parlance. In other words, it would cost healthcare systems not to prescribe the intervention. But in terms of getting to your question, it really does depend. In the other areas like retina, because it's a rarer disease, there I think it would be more on demand, and we would be able to, I mean, as long as we are in the position to produce batches economically and lay them down, that's fine. But it will depend on the centre.
You don't want too long a shelf life because you want them to replenish it, won't you?
Well, that's true, but I mean it's driven by patients. I mean the whole model will work on a patient by patient basis, so it doesn't necessarily matter. We'll price the products on a per patient basis or a per dose basis. So if they want to buy doses in bulk, then that's fine.
Any other questions? I've got one. On your phase 2 stroke trial, is there just one initial injection or whatever treatment you give people, or is it a serious?
Yeah.
Or is it serious? It's just one.
No, it's a good question. In both cases, actually, these are one-off treatments, and actually that's fairly typical in the field, and actually raises another issue of reimbursement. So generally, most stem cell treatments are envisaged as single one-off treatments, which come at a relatively high upfront cost. Yeah. To garner significant benefits over time, especially where they're addressing chronic diseases, as these ones typically are. So how's a payer going to reimburse you for that? And I think what we'll see, rather than sort of just paying you for the drug up front, there will probably be annuity-based payment systems and payment for performance. So if you can show follow-up in treated patients and a sustained effect, then you'll be paid for that over time, rather than asking, NICE to recommend a treatment that comes at a vast upfront cost, and the potential effects of that treatment over time can't be evaluated in that particular patient.
Pay on performance almost.
Exactly, yeah. Or at least we have to be prepared for that.
Yeah. And how quickly, and once you've had that treatment, would somebody see a reaction or a benefit?
Well, in the trials we've done so far, it's usually manifest within 3 months and sustained out to 2 years. It does vary, and especially in stroke, because every stroke patient presents somewhat differently. But generally, if we're going to see anything or anything meaningful, it will usually be manifest within around 3 months.
So hence your trials take as long as they take. They're not just simple a few weeks and you get a reaction.
No, absolutely not. And in some cases you do see very early reactions, but in most cases you've got to discount that as a placebo effect. Okay. So you've got to monitor patients for a reasonable period of time thereafter to discount any placebo effect. And over 2 years it would be very difficult to attribute that to a placebo reaction.
Gentleman down here at the front.
In diabetes, by which mechanism do you want to help the patients? Are you going to regenerate non-existing or very poor blood vessels or the tissues or the skin?
So essentially in lower limb ischemia, primarily it's driven by loss of microvasculature actually, and what we see very clearly certainly in the animal work we did, and it's very easy to see this because you can image muscle fibers very straightforwardly because you're not working the brain, so things are a hell of a lot easier to do. And what we saw was vastly improved capillary density in these hind limb models of limb ischemia. These were rodent models. So the idea is that you stimulate microvasculature sufficient to be able to essentially improve blood flow sufficiently to either avoid amputation, that's the holy grail, or at the very least enable those patients to be eligible for revascularization. So surgical revascularization is the current standard of care for critical limb ischemia patients. But many of those patients just won't be eligible because of their wound healing problems with diabetes and so on. So we believe that the treatment at the very least should be able to render more patients eligible for that and thereby avoid amputation. But what we'd really like to see and what we saw in the animal models very clearly and dose dependently was this improvement in vasculature sufficient to restore blood flow right to the extremities. And if we can do that in man, that will avoid dry gangrene and we hope ultimately amputation.
And final question on the other side of that row.
Can any of the programs work in a preventative manner? In other words, before actually the person
No.
gets the stroke?
No, and the reason for that is, I mean, it's very difficult to conceive of a treatment that would preempt a stroke insult. I would say you've got to lead a better lifestyle or a healthier lifestyle to do that. But if you don't do that, or you're unlucky enough to do that and still get a stroke, then that's where the cells come in. Similarly, in the case of the blindness disease, that's an inherited condition anyway. So unfortunately, if you have a family history of RP, then you have a much higher chance of actually suffering it anyway. So now, I mean, CLI, again, that's a classic lifestyle one as well. I mean, it's very common in diabetics. Around 70% of diabetic or CLI patients are diabetic. So again, improved lifestyle will avoid the need for any of these treatments, you know, in time as well. But the way things are at the moment, an aging demographic and the ever-increasing prevalence of obesity and diabetes is actually increasing our markets, if you will, in these conditions. So,
Thank you.
Michael, thank you very much indeed. Thank you.